Hello Year 11 Triple Biology. This is going to be a very short lesson, this one. It's on serial dilution. You will need your microorganisms and their application booklets in front of you, and you need to turn to page 21 for me, please. So the lesson objective today, we've already carried out a little bit of this work in a previous lesson, is looking at understanding how to carry out what we call a serial dilution. So on page 21, you have this image, albeit a black and white colour copy rather than a colour copy. If we'd been in school, I would have got you to do this with some food dye. So you could actually see the dilution actually taking place, and as the colour of the solution starts to get paler. So what is a serial dilution? Well, it's a succession of step dilutions, each with the same dilution factor, where the diluted material from the previous step is used to make the subsequent dilution. So for example, you might want to dilute a stock solution, an original solution, by half each time, or you might want to dilute it by a factor of 10 each time. These are the two most common that you will come across at GCSE. This allows you to make a wider range of concentrations than a standard dilution. 1 cubic centimetre of the solution would be added to 9 cubic centimetres of distilled water. Remember, distilled water has no other nutrients in it, okay? It is as pure as we can get it. And this is repeated in this case, okay, in this example, 4 more times until the 5th test tube, this last one here at the end, is 100,000 times more dilute, okay? So we would say that in this dilution, taking 1 from the original sample, okay, and putting it into 9 with another one, we would have, this would be 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4. So it looks like an 8, 10 to the minus 4. So we do this with bacteria in suspension, and from our previous video we said the reason why we do it with bacteria in these solutions is because in the original solution there might be so many bacteria that when you pour it onto the agar plate, it just becomes almost like a white creamy mass of bacteria. You cannot count the individual colonies. So by diluting, by carrying out a serial dilution, we know exactly how many times we have diluted it. So say this was the tube that we were going to pour out onto the agar plate. We would count the number of bacteria that had grown on that agar plate and be able to work out how many by going back the other way. So instead of diluting, going back and concentrating, how many were in that original tube, okay? So, in practicals, you're normally told what factor to dilute a solution by, and then you're normally given a diagram to complete before doing your dilution. I've got 3 examples on this page. It starts off with, or 3 examples over the next few slides. Starts off with a really easy one, and it does get harder. Believe me, even I had to check my answers with Mrs. Lees and make sure that I had it right. Okay, so please do not panic if you don't get this at first, okay? And do not panic if example 3, it's starting to confuse you, okay? I'll try my hardest to explain this as clearly as I can. So I want you to complete the diagrams, right? And then you would normally carry out this serial dilution using a fake stock. So I would give you a fake stock of this 4% glucose solution that we're calling G, okay? And normally in our fake stock, we give it some food dye, some kind of colouring in there, so that as you dilute it, you can see what happens. And each case you're going to be diluting it with your distilled water. So G is the glucose solution and W will always be distilled water. Okay, so turn over the page to page, where are we, 22 for example 1. So this is a nice easy one that we're starting off with. So you're now required to carry out a serial dilution of a glucose solution G. And we want to reduce the concentration by half between each successive dilution. So if you needed to make up, let's say, a glass of squash that was half concentrated, you would measure out half the amount of squash, exactly half the amount of water, and you would add that to the squash and you would have a half diluted solution, okay? Whatever the solution concentration was in the first place, it would be half. If I wanted to make that squash halved again, then I would take half of that diluted squash and then I would add it to the same volume of water, and that would dilute it again. And you could keep going until your squash becomes so pale and tasteless, okay? It is more water than it is squash, okay? So in this first beaker, this is what we will call beaker number 1. We've got beaker number 2, beaker number 3, and beaker number 4. It wants us to complete to show how we'll make 2 further concentrations of the glucose solution G here. So we'll start off and have a look at what they did in the first step. So in this beaker, we've got 30 cubic centimetres of 4% glucose solution, okay? Bear in mind that cubic centimetres, we can use millilitres, okay? They are interchangeable. 1 millilitre, 1 cubic centimetre, they're exactly the same thing. So there's a little video that I'm going to post for you later, and I think he talks mainly about millilitres. So we've got 30 cubic centimetres, and the solution strength is 4%. We want to take half of this. 15 cubic centimetres is half of 30. And we would use normally a pipette, clean pipette each time so that we're not contaminating our solution. And we would pull up, let's say, 15 cubic centimetres of this 4% glucose and put it into this beaker. So it would go in here. In this beaker, there would already be 15 cubic centimetres of distilled water. So just as I described with the squash, we are diluting this by half. Because we're diluting this glucose solution by half, and then we would stir it, mix it, okay, give it a swill, okay, to make sure it's evenly distributed. Because it's diluted by half, it's now down to 2% glucose solution. So, how do I get it half again? Half of 2 would be 1% down here. So I want to make a 1% glucose solution. Have a little think about it. If you think that you can do it yourself, can you write on what you would put into this beaker to make 30 cubic centimetres, that's the volume I want, of 1% glucose solution. Pause the video and have a go yourself. So hopefully you have figured out that you would take 15 cubic centimetres from the previous solution, that's the 2% glucose. So we want 15 cubic centimetres of 2% glucose. I'm trying to write as neatly as I can on here. We would add it to this beaker, and in this beaker already there would be 15 cubic centimetres of water. I'm not going to write distilled water, just put water, but you know I mean distilled water, W. So we have halved the concentration again in this beaker 3. If I want to halve the concentration again a further time, then instead of it being a 1%, it's going to go to 30 cubic centimetres, that's our volume, of what's half of 1, a 0.5% glucose solution. So we want a very weak glucose solution in this beaker 4. Again, pause the video. See if you can write those on yourself, and then I will go over the answers. So hopefully you've come up with the idea that from beaker 3 this time, you take 15 cubic centimetres of 1% glucose solution, G, and you add it to this beaker that's already got 15 cubic centimetres of water, W. So I'm hoping everybody has followed that. So this is making solutions that are halving each time. So I've underlined that there, okay? We want to reduce the concentration by half. So that's our first example. Turn to the next page. So the next page is 23. And this time we are going to make a serial dilution. But this time, rather than halving, we're going to do diluting it by a factor of 10 between each successive dilution, okay? So we're going to start off, our solution is 0.3%, and we're going to call this solution X, okay? So they have put in this beaker 10 cubic centimetres, okay, of the 0.3 solution X. There's no water in there at all, it is just purely the 0.3 solution. We want to make a serial dilution, as we said, by a factor of 10 between each dilution. So we want this one to reduce by a factor of 10, reduce by a factor of 10, reduce by a factor of 10, until this solution is quite a weak solution. Each time we want to prepare 10 cubic centimetres. So whereas previously we had 30 cubic centimetres in our beaker, this time we want 10. You should use the beaker shown to show you how you prepare the serial dilution, and you will need to use 9 cubic centimetres, so it's giving you a clue, of each concentration of X in the investigation. So we are going to take 1 cubic centimetres of the 0.3% solution X. We're going to put it in this beaker. We need, okay, to have 10 cubic centimetres in here. So how much water? You should be able to guess that that will be 9 cubic centimetres of water, and we'll call water W, okay? It would be distilled water. So in here, we would have, okay, 9 cubic centimetres for our volume, okay, of 0.3 times 10 to the minus 1%, yeah, because we've diluted it by a factor of 10. So if we wanted to write what this percentage actually would be, if we're doing 0.3 times 10 to the minus 1, it would become 0.03%. So very, very weak solution. So in other words, this is a tenth of the concentration of the original solution. Or we could say that this is a 1 to 10 ratio here. So there are various different ways that you can show this. So pause the video, see if you can fill in the next two beakers, so diluting them by 100th and by 1,000th, okay, for the last beaker. So if we call this beaker 1, that's the original solution. This is beaker 2 that's been diluted by 1/10. Beaker 3, I would like you to dilute that so it's by 100th. And beaker 4, this one is going to be by 1,000th. Okay, pause the video, have a go. So it is really this simple. All we're going to do is we're going to take 1 cubic centimetre from our beaker 2, our 0.03% or 0.3 times 10 to the 1. 1 cubic centimetre of 0.03% or the 0.3 times 10 to the minus 1. We're going to add to that solution 9 cubic centimetres of our distilled water. So now we'll have 10 cubic centimetres. Actually, that's where I made a mistake there. 10 cubic centimetres. 10 cubic centimetres of, this time, 0.3 times 10 to the minus 2%. So what is 0.3 times 10 to the minus 2? Get the right number of zeros here, 0.003%, okay? For the last beaker, the 100th of the original solution, we're going to take 1 cubic centimetre, this time of the 0.003%. Sorry, it's very squished on here. We're going to add 9 cubic centimetres of water. And this time we should have 10 cubic centimetres of 0.3 times 10 to the minus 3%. And that should be 0.0003%. Right? So this is 1 to 1,000. So it's 1,000th, okay, diluted. This is 1 to 100. And this is 1 to 10, okay, a dilution of 1 to 10. So hopefully that's made sense, okay. Pause the video, replay it if you're not sure, okay, and go through it so that you understand how to make factors of 10 dilutions, successive dilutions. Now, I said the last example was the hardest one. It's not super hard, but it's because of the volumes that they're using. It might just confuse you a little bit. So it does take a while to get your head around it. Believe me, both myself and Mrs. Lees looked at it, and it took us a few moments, okay, to figure out what was going on. So this time we are starting off, rather than with 10 or with 30, we're starting with 20 cubic centimetres. And it is 10% this sodium chloride solution, and they're calling it solution S, okay? So this is what we're going to be diluting. It wants you to dilute this sodium chloride solution by a factor of 10, okay? We're only going to do it, we're going to do it once. I'll do the first one as an example, okay? And then we can imagine that we're doing another 2 beakers like we did on the previous page, okay? A beaker there. So we're going to call these beakers 1, 2, 3, and 4. You might see them called A, B, C, D. Okay, it depends how a scientist is setting it up. So we're going to prepare, in each beaker, we want there to be 20 cubic centimetres, okay, of solution. And we need to complete the figure 1.1 to show how we will dilute it. You can use the 2 beakers shown, and then it says draw as many beakers as you need to prepare sufficient concentrations for an investigation. So we're just going to go up to beaker 4 here today. Under each beaker, state the volume and the concentration of the sodium chloride. Use one arrow with a label above the beaker to show the volume and concentration. Use another arrow to show the volume of water added to prepare the concentration. So, in the previous one, to do a dilution by a factor of 10, we had 10 cubic centimetres and we took out 1 cubic centimetre. This time we've got 20 cubic centimetres. So instead of taking out 1, we want to keep the ratios the same because it was 1 to 9, 9 of water and 1 of the solution. This time we're going to double it up, so it's going to be 2 to 18. Can everyone see the ratios? It's exactly the same, it's just been doubled up our amounts because we're making 20 cubic centimetres, not 10, okay? Otherwise, if we do the 1 to 9 here, we're not going to have enough volume, we're not going to have our 20 cubic centimetres, okay? So we want to keep it at this. So we're going to take from this original solution 2 cubic centimetres, okay, of the 10% sodium chloride. I'm going to put NaCl just to make it easier. In this beaker already, there will be some water. What is the volume of water? It's not going to be 10. It's going to be 18 cubic centimetres of water in this beaker. That's going to make our volume 20 cubic centimetres. And because we've diluted it, it's 10% times 10 to the minus 1. So 10% times 10 to the minus 1 is going to be 1% solution. Yeah. So we've reduced this by a factor of 10. So we've taken 1/10. Okay. If we imagine our beaker number 3 that I've drawn here, we're going to take 2 cubic centimetres, this time of our 1% sodium chloride. We're going to add 18 cubic centimetres of water. So we've still got 20 cubic centimetres in our beaker, but this time we've got 10% times 10 to the minus 2. So 10% times 10 to the minus 2 is 0.1% solution concentration of sodium chloride, okay? So this will be 1/100th of the original solution. In our final beaker, we're going to take out 2 cubic centimetres of 0.1% NaCl. There we go. We're going to add in our 9 cubic centimetres of water, giving us a total volume of, what am I doing? Not 9, apologies. I knew I would make a mistake. 18 cubic centimetres of water, and we would have a total volume of 20 cubic centimetres, this time of our 10% times 10 to the minus 3. 10 to the minus 3, 10 times 10 to the minus 3 is 0.01% concentration solution. So this is 1,000th of the original solution's concentration. Hopefully that has made sense to you, year 11. If it hasn't, please do ask questions, okay, in the Teams feed, or you can send me an email separately, okay. I don't want you to panic too much about this example. This was the hardest one. As long as you understand example 1 and example 2, I don't think they're likely to give you anything more complicated than that. However, if you are going on to study A-level, you do need to know how to do this, okay, because you will be making up solutions, okay, to certain concentrations for practicals, and this is what our lab tech, Mr. Hancock, does normally on a daily basis when we're in face-to-face teaching. If I want glucose solution, or if Miss Lees wants a sodium chloride solution of a certain strength, he will be required to make this up for us, okay. So nice short one for today. Make sure everything else, okay, previous has been done, okay, and we will leave it at that today. Have a break. Thank you, year 11.